So I'm Darcy Peterka. I'm a senior scientist at the Zuckerman Mind Brain Behavior Institute at Columbia University. I'm kind of a jack of all trades person. My background's math and chemistry, but I shifted it to neuroscience now about 14 years ago. And I generally develop tools that let us read and write neural activity and then try and use them to solve actual, real biological problems. Like Andrea said, please abruptly interrupt if you have any questions. I really, I am someone who very much prefers in-person interactions and kind of conversational style talks, which you'll see. Hopefully it works reasonably well over Zoom. And I have a lot of material, but I'm happy to take it as fast or as slow as you guys want. So please interrupt me if you have any questions. Extra credit to anyone who can identify the movie that this was.
The Day the Earth Stood Still.
Bruce gets the win. That's a great flick. Anyway,
gray hair. Yeah.
Yeah. So if you look at Homer Simpson in this slide, what do you think? What's he doing? Yeah. Presumably he's thinking and he's kind of looking. And that makes me think of, what we really try and do. And this is a nice Yogi Berra-ism. So Yogi Berra was this baseball player who said a lot of things that sounded really simple, but actually carried some wisdom. And the statement is, you can observe a lot by watching. And that's really what we do in most of our experiments in neuroscience, at least the community here is what we want to do is we want to see what's going on in the brain while the animal is doing something. And we want to use that and learn and try and figure out what's going on and what kind of computations are going on. And this is sometimes a challenge because the signals aren't always as clear as we'd like them to be. And we're trying to sometimes measure things that are really small on top of things that are really big. So, there's a slide that I like and say, what's our job in functional imaging? And our job is really measuring grass, right? So imagine we're down here observing something and we want to measure grass at the very top of the building. It's really, really hard to do that. And the reason it's really, really hard to do that, if you have a background signal that's, sorry, my screen just froze. I'll see if it comes back. My apologies. Maybe I have too much going on. Hopefully yours didn't freeze.
It's okay, yeah.
Yeah, okay, perfect. Anyway, so what we're trying to do is we're usually trying to see a small signal on top of something big. So generally what we want to do is we want to cut down the background. Imagine trying to see the grass on top of the Empire State Building. We'd have a tough time saying how tall it is compared to the height of the building. So usually what we do is we want to remove the Empire State Building, remove the background and lower it to the point where we can see the kind of the height of that grass. So almost all the imaging techniques that we use, whether it's two-photon fluorescence, whether it's the indicators we use, whether it's whatever, the goal is essentially to remove the background as much as possible and let us see the thing of interest as close as possible. And what you see in those little blue bars, that 0.1% and that 100%, that's actually numbers saying how many photons would you need to reliably detect a signal if you had a signal modulation that's 0.1%. If it's 0.1%, you'd need 16 million photons to detect a change reliably over the noise. But if you could somehow remove almost all the noise and have it so the modulation change was 100%, you'd only need to collect 16 photons to reliably detect a signal. So that goes to say that almost all our imaging methods and everything I'll talk about is trying to find ways and essentially to remove this background so we can measure signals. So practically, what do we do? We were all used to seeing videos like this. I think this is a mouse that's been trained. This mouse, when it sees vertical bars, it knows to lick. It's a very decent mouse. It knows, you'll see its tongue start flicking around. It does it reliably and kind of day after day, session after session. So we have behaviors. We have the mouse doing some kind of perceptual observation and computation in its brain. And then using calcium imaging, we can image. This case is imaging in V1 while the mouse is doing the task. And our goal is to take those activities and turn them into behavior. And usually we have field of views that look something like this. And, you know, we want to know what's going on. So we say, this is great, right? The first time I saw calcium imaging of a behaving animal, I mean, I was amazed, right? I'm looking at the neurons. I'm looking at the mouse thinking. It's doing something. But, you know, how do we figure out what this actually means? So we only witness a small part of the brain. We try and regress it against something else. We try and figure it all out. And it's hard. And one reason why it's really hard is the brain and the behavior of the animal is much larger than what we're observing, right? I'm only imaging a very, very small part in mouse visual cortex. The mouse is doing all kinds of things related to its posture. It's maybe daydreaming. It's doing lots of things besides my task. So it becomes a challenge. So one way we thought we could get rid of that challenge is we really like to do the things that Andrea had mentioned earlier, which is to kind of close a loop. We'd like to be able to link neural activity with behavior and then make predictions and test it. And how would we do that? So in this case, right, we have a behavior that we like with a mouse does some behavior. Do you guys see my pointer, a laser pointer on your screen?
Yes. Yes.
Okay. That's a big delay I see. When I do it on mine, it freezes a screen. So I may not be able to do it effectively. Yeah, it did it. I'm doing an experiment here. So now I know what definitively causes my screen to hang. So I won't do that anymore. N equals 2 learning. I'm not that bad. I'm not too old yet. I apologize. Let me see if using the Zoom annotation would actually be more helpful for this. My apologies. I came into work so I'd have high bandwidth so my kids' Zooms wouldn't interrupt the talk. And here I am, thwarted.
Darcy, I don't know if you have Dropbox going. I found that Dropbox was interacting heavily with my presentation. I don't know if it's something that might happen to you too.
I can certainly kill Dropbox. I have like 100 sharing things. At any rate, I think I can do it with just the annotation by just having a mouse cursor. Can you see that on the screen?
Yep.
You see the mouse on there? I mean, not the, you know, my little pointer.
Yeah, I see it.
Okay, yeah, 2. Okay, fantastic. Okay, hopefully this is the end of my experimental glitches. So at any rate, we want to see a behavior and we want to do observations. The observations we generally do are 2-photon imaging, though we do some 1-photon imaging as well. We have to take those measurements and then we have to map them. And by mapping, I mean we have to figure out which cell seems to be involved in what part of the behavior. And then we'd like to be able to modulate those cells precisely using a deterministic system where we can turn individual cells on or off at our leisure and see if that ends up changing the behavior. And then if that changes the behavior, or if it doesn't, then we refine the observations and we iterate this and we go around and around in closed loop. And that's generally what we mean by when we say, if you want to be looking at the behavior, figuring out which neurons are active, and then controlling or driving the experiment based on what we see. And my talk will be not so much about the behaviors, but will be much more focused on figuring out good ways to do the measurements and the mapping. One thing that's become clear in the last few years is imaging large regions and bigger areas of the brain has become more straightforward. So, you know, the middle picture is kind of what a standard system used to do a few years ago. And it could image maybe like a few hundred microns or 500, 600 microns by 600 microns. And that was about as good as you can get. But through a bunch of work predominantly by people like Spencer and many others listed below, now we can image these larger patches. So on the right side of this, you see one of my microscopes that has about a 2 millimeter field of view, and it can image many more neurons. And this is becoming actually pretty easy now. I mean, Thor Lab sells a mesoscope. There's many companies that sell mesoscopes and you can build them. And having access to all these cells is great, but it makes analysis harder and it comes around to some other problems. And the problem is that even when we image these areas, as imaging large areas with high speed and high resolution, it's still very, very hard. And the reason for that is something that, I think people should be able to figure out. Could anyone tell me why you think it'd be hard to image big areas with high resolution and high speed?
Well, you're limited by the size of your point spread function, right? And if you're doing two-photon, so that's the resolution. And if you're doing two-photon imaging, it's a line scanning system. So the larger your field of view, the slower it's going to be.
Yeah, that's exactly right. Multi-photon images are generally made from pixels or voxels. And your pixel rate, you can just compute it. It's a number of rows times a number of columns times a number of frames that you want. So then you can say, well, how long do I hang out at a pixel? Well, delta t is just 1 over that multiple. The trouble is the signal is generally proportional to the dwell time of each one. So you could go faster if you had an imaging system that could move infinitely fast, and you'd spend 1 over infinity at each pixel or 0 time, you wouldn't get any signal. So there's these fundamental trade-offs generally on these systems between having high resolution, high pixels, and high speed. And what people should be thinking about now is when we see these is, well, sure, this is what we have, but is this really what we want? So when we think about it, we like to say, well, do we need all the pixels? And do all the pixels really need to look like this? And this is that little dot that looks like the two-photon, for Americans, football, for, maybe Australians, rugby balls, whatever you want to call it, right? And the truth is, no, it doesn't need to look like that. It really depends on what you want to do, what the task requires. And a lot of these things were driven by some just fairly straightforward observations that I think, many people made, which is if we actually look at images of the brain, at least in cortex, you see that the images are generally not so dense, right? So if I click this, these are sequential imaging. I'm just using an electrotunable lens and I'm stepping through frames. I'm displaying them all at once, but we can see different activity at different depths in Z. And on each one, you see it's generally a dark background and you have a bright pixel, all right? So if I just say, forget it and just do a simple kind of, thought exercise, and I look at these images on the bottom, all I'm doing is I'm taking the images at the top and I'm literally doing addition in image J. I'm not doing any imaging tricks to do it. I'm just saying, well, the thing that's labeled 1 + 2 is literally that. It's the image at 165 added with an image from 195. I keep on going, and you can see even in the last image, it combines multiple images together, the 1 + 2 + 3 + 4. There's a little more background, but in general, you still see neurons firing, right? We still see them as cells. It's not all completely blurry. There's just a bit of a high background. So we kind of observed these things and said, well, hell, why do we care about having a single point spread function if we know we can essentially add these things together and generate images that might generate useful signals? So the thing we used to do this, and thanking Nico for his beautiful talk yesterday, I don't have to spend much time talking about spatial light modulators, but we said, okay, we can do it, but how do we generate multiple spots in space? And the device we choose to use are these spatial light modulators, which are these, at least as we use them, a liquid crystal on silicon device that changes the phase of the light going in. And by changing the phase of the light going in, we can change the nature of the spots that we generate. So what you see in this picture here, you see the very top part of it supposed to be the representation of basically a planar wave. If I have a plane wave, it goes through an objective, and then it gets focused to a single point. But I can do all kinds of things. I could put a tilted wavefront on it, and then it shifts the beam over sideways. I put a tilt on the other way, and it shifts it in another direction. But I can put a lens function on, a spherical wave front on it, and then I'll change the focus. I can make multiple spots. So now I have a device that I can flexibly control the phase of the light coming in, as Nico was talking about yesterday, and I can generate multiple spots in space. So this provides me, these tools give me ways of controlling the light field in a way that's, one, determinable by the user within the rules of Maxwell's equations, and three, very, very flexible. If I wanted to switch from one pattern to another, all I have to do is send a signal to a computer. I don't have to take my microscope apart, move an optic, or do whatever. So we decided to build microscopes using these tools to do kind of enhanced, faster imaging. And one thing I'll note is I use a lot of my experiments in this talk, mainly because I know them well. It doesn't mean that my experiments are necessarily the best ones. It's just the ones that I know. So there's other people, and I'll show some slides later on showing that people use similar techniques as well. And I'm not going to be highlighting them because I don't want to give a review. I want to just kind of give it there. But I certainly don't want to pretend that I'm the only one thinking along these lines. So for this experiment, this was work done by a postdoc, Wei-Zhen Yang, who worked with me and Rafa Yusta, and now he's an assistant professor at UC Davis. And he took a normal 2-photon microscope. You have your ultra-fast laser, some power control, and then some galvo mirrors and so on. And he attached a SLM module to it. And these SLM modules are essentially a telescope to expand the beam to fill up the SLM, and then another telescope to relay the SLM's the focal plane to the back aperture. And by doing this, he was able to then use the SLM to basically generate 2 beamlets. And these 2 beamlets are dispersed axially in space. And then he's able to scan over a region. And when you get that, since I'm not doing any tricks with any kind of temporal multiplexing here, what you get when you scan those is you get a superposition of both images that I scanned, very similar to the sum, the arithmetic sum that I showed you before. So if I have objects A and B on each page, my combined image will be A superimposed with B like it is there. And again, if anyone has any questions, feel free to stop and ask or whatever. So, with this microscope, you can generate these signals. So what Wei-Zhen did is he said, well, I want to look what's going on in layer 2, 3 and layer 5 at the same time. So he generated 2. Well, first what he did is he took an image just at 170 microns. What you're looking at is the standard deviation time series projection of a movie. So, right, the cells that are bright are the cells that were kind of active during that time. At 170 microns, and then he took another movie at 500 microns. And you can see they look different. They're color-coded just arbitrarily for simplicity. If I just took these time series together and added them, what you see is these neurons would be interspersed, right? Looks a little bit messy, but not horrible. And if I, instead of doing it by doing an arithmetic sum, I instead just turned my SLM on and projected 2 spots simultaneously to the upper and lower plane and scanned them simultaneously, I generate an image that essentially looks exactly like the sum. And some of you might be a little bit worried about this because, if our goal is cellular resolution, like we just kind of made a mess, right? We just sandwiched cells on top of one another. So what do? You guys should all be able to answer this after Andrea's talk. What do we need to do? Do I just circle ROIs like a dummy?
Time and space correlation.
That's right. I need algorithms to fix this kind of stuff. And it turns out it's actually no problem. It works really, really well. And, when we did this paper, we collaborated with Eftichios, an old collaborator of Andrea, and Liam Paninsky, to also work on source extraction algorithms that are able to disambiguate overlapping sources. So what you kind of see in this picture, when these ROIs, the regions of interest, are overlaid, there's a lot of overlapping regions of interest. But it's no problem. If you have a great algorithm like the CNMF and you like Cayman or these things, you can actually do a lot. So we showed in that paper that, whether we did sequential imaging or simultaneous imaging, the types of signals and the SNR we generated was the same. We were able to image lots and lots of things. It looked really great. And, so one problem kind of solved, right? How do we get more neurons when we need to image to understand what's going on in the brain? Well, we just collect more neurons from different areas and use essentially algorithms to help sort it out. So normally I would spend some more time on this, but of course, Andrea says that's such a great time. I'm explaining this and showing what it is. I'll just basically highlight a couple of things just to reinforce it, but I won't go through it in detail, right? what we collect when we collect an image is we collect essentially, if we vectorize a 2D image, that's similar to what Andrea was talking about for the memory mapping, right? We just take instead of having a row, we just string it together. So one row, and then instead of drawing it here, we just put that row and move it here and just essentially make it one long vector. So that vector has dimension d, which is the number of pixels total in image. If you had 512 by 512, it would be 256,000 pixels, let's say, d. And then you have time. The time is essentially just how many frames you have, right? And we want to factorize that. And we say that what this is equal to some spatial matrix times this temporal matrix. And this is this outer product that Andrea was talking about. So this says we believe we can separate these movies into something that tells us about some spatial footprints of objects and then the temporal activity of those things. So if we look at these matrices, like what they really are is a row of this temporal matrix is essentially the temporal dynamics of neuron I or source I, if it's an axon or a dendrite or whatever. But so essentially each of that is a clean trace of just one of the objects that's generating signals, right? If I look at the column of the spatial matrix, the column is something that's just in pixel space. So if I have a number in somewhere in that column, it's telling me that some of this neuron sits in this pixel. So essentially this column is a spatial footprint of the neuron. And you can see my little crappy line art representations next to what I say. So like, right, each column of A is a spatial layout of each source. Now, there's something else that this can allow for, which is the row of A, in a sense, that characterizes the weights of the contribution from different sources. So those weights are saying how many neurons are mixed. So when I have two neurons overlapping, then there's some pixels that have weights from two neurons. So that's essentially what the layout of this is. The rows, you do a factorization problem and you can do it. And of course, you can do some math on here and you say the total fluorescence is equal to the sum of A and the C. That's if it's noiseless. If it's noisy, you can add some noise terms. And then what you do is you can do these giant, you can essentially do optimization. And if you do it well, you can factorize this. And this was made very efficient by Sebastian Fang's kind of development of non-negative matrix factorization. For those of you who know Sebastian, I think he'd probably shoot me, but I think NMF is probably the most valuable contribution because it can be used for factorization in many, many, many different instances and really enables a huge class of problems. So, this NMF method lets you do an efficient matrix factorization that lets you separate out the sources. And just how Andrea showed it in the previous one, what you see on the bottom are individual neuron templates that this algorithm selected, the spatial footprints of each of these things. And these things look really nice, right? I mean, they look like neurons. And this happens in a sense in an automatic fashion, right? So these kind of tools let us know that if we design hardware with the software and signal extraction algorithms in mind, we can essentially do imaging that's smarter than if we just did one independently of another, right? If I just mixed my signals together and I had no idea how to separate them, I'd be in trouble because I would just have, essentially I'd have more signals, but it would be a mess. I would never be able to disambiguate them. And now if I had CNMF or an algorithm like that, and my images were so simple that neurons never overlapped, I wouldn't need it, right? So in a sense, it's by taking advantage of both of these tools, both the software and the hardware, that lets you create these kind of imaging systems that is great. So I think this is really a fundamental difference in the way people have started doing imaging in the last few years, which is in the past, people would spend a lot of time trying to make sure that their signals were never contaminated. They would try and make it as clean as possible. But with the advent of these kind of algorithmic tools that we had heard about this morning, is we can actually change the way we do our experiments and purposefully mix signals together and then rely on the algorithm and some priors and so on to help extract out the signals of interest. And that's a really powerful approach.
Okay, so Darcy, I have a question about this because I understand the premise behind how source extraction works. And essentially in this experiment, you have one PMT And I understand how the experiment is done and how the data from the different layers is put on top of each other. But let's say you were just imaging at 170 microns and you were just looking at that one plane. You would still have cells that might overlap at that one plane. So how does the algorithm know? Like I understand how it would identify what different sources are using CNMF, but how does it know what layer each of the different sources are from? Like, are there priors? And because at first I thought perhaps the deeper layer is because it's deeper, it's less bright, but because of the way that you're collecting this imaging data using the SLM to scan both at the same time, that's not the case. So how does the algorithm know how to do that?
No, that's a great question and a great point. So the algorithm doesn't care. The algorithm on its own does not care at all about where the neurons are in space. That's something that, of course, we as humans care about for the experiment. So what we do, what I kind of glossed over, but it's in my little slide notes, is we have prior information, right? Because what I can do is I can separate the task of generating an anatomical image from generating a functional activity image. And that's generally the case with neurons because neurons generally don't move in space, right? Or at least not much. So what I can do is in this experiment to kind of supercharge the algorithms, what I can do is I have the individual images that I run, just one plane at a time. I collect data operationally for a few minutes at each one. And I just run source extraction on that to get an anatomical prior. And then I can use those priors as a seed for the algorithm on the whole thing. Now, if I only cared about signals, I don't have to, but if I want to correlate it to anatomy, I go back to using priors. And that's actually another part of this kind of, these tricks of mixing is also separating the tasks that we want to do and realizing that we can separate the task of understanding the anatomy from the functional recordings. We can do that in 2 steps in the case that the anatomy doesn't change much. This may not be the case for imaging where if people are imaging microglia and they're moving around in the brain or something like that. But generally for neurons, astrocytes, and other things where the change in position is slow relative to the type of imaging, you can separate these out into distinct phases. And that gives you a lot of advantage.
Awesome, thank you.
No problem. And
also
then,
a question, Darcy. Did you try with more? And when does it break? So when you did the experiment, you were using viral injections, right?
Yeah. Yeah.
Because
so I would say my next slide might answer some of that.
Okay. Sorry.
So I mean, like, so the question is always, how much more can you do? And, we've done a lot of things saying, well, like, how fast can we switch the device? We can switch between layers in a few milliseconds. And then we said, well, how can we switch between layers and generate multiple layers together and do a bunch of imaging? And this is like multiple planes on multiple things. And then we started doing slicing like this, where in this case, we're imaging 3 planes at once, and we step through them in a few milliseconds. But we've done experiments where we've done up to about 13 planes at once and extracted out useful signals. And I'll actually talk about this more in the coming slides. And the limit of that has to do with some fundamentals about signal collecting, right? So when I add these signals, in an optimal situation, the signals at each plane roughly have the same intensity, because that means that the background from each plane is about the same. And, going back to what we know about collecting photons or counting, anything where we have counting noise. So if we have counting noise, the number of photons collected is n. The variance on that is the square root of n. So if you imagine as I add more and more planes together, unless those planes are completely dark, they're adding background. That background has some natural variance, which if it gets high enough, it'll obscure the real signal. So the limit on what you can actually do is more given by the complexity of your sample than just the technique on its own. So if you had very sparse expression on multiple layers, you could probably do every one. You could do 20 layers and just be fine. In fact, Ali Pasha just threw up a preprint that they think they did yesterday, that they did like 25 layers or something like that simultaneously, right? If the layers are somewhat sparse. If the layers are very, very dense, then it's much harder to do that because all the backgrounds add up, and eventually the variation in signal of your background will swamp small individual transients from calcium. We liked this approach when we did it. We tried some vessel beam imaging, which we didn't do it that well. And with a virus injection, the background is too high. So we shifted to these multiple beamlets because we could control essentially how much signals we want to mix. We could say, oh, we have a lot of complexity, let's mix 2. If we have less complexity, let's mix 6. And we can just change that on the fly. So the limit is more given by the background than the fundamental optics. So I should say is like, what do we think about these approaches? Why do we really care about these strategies again? And it's really just a change of the way that we think about imaging, which is that these kind of, how do we get more signals and things that we care about and think about the things we can control? So if you look at this expression, this is, I think, the only real math I show in this talk, right? I mean, I glossed over the matrix factorization because Andrea already did that. And this is basically a thing that tells us about the signal, the fluorescence that we collect on our microscope. And the first term is a collection efficiency phi of the collection. We have a square 1/2 because this is on a per photon and we have a 2-photon process, whatever. So this is a fluorescence collected per photon in theory. We have a phi of F, that's the fluorescence quantum yield. That tells us how much every time a fluorophore is excited, what fraction of it emits a photon, right? When they excite GCamp or GFP, most of it goes off as fluorescence, but some doesn't, right? Then I have my 2-photon cross section that's kind of given by nature. And then I have some term that's this double integral of C times this I squared dV. And what this term is, the C is just a concentration or a location of the fluorophores in my sample. And the I squared dV is essentially the shape of my point spread function. Where is my light? This basically gives us an expression that says, how can we relate signals to a bunch of things in the world? And the total signal is there, that's fine. The quantum yield and 2-photon cross-section, those are given by nature. I have no control over them, right? I mean, people who work in protein engineering have a little bit of control over them, but I don't as a user. I just get the probes and I try and make it work. But what I do have control over is this term, which is the spatio-temporal distribution of light. So a lot of these approaches, like the ones I just showed, the ones I'm going to show shortly, I say, we're going to try and improve imaging by changing the spatio-temporal distribution of light. So normal 2-photon imaging is the extreme case where you want the smallest, most precise spot possible. So in some sense, it's ideal for imaging very dense and complex samples. You have a small delta function that's sampling an image. So you get the kind of a somewhat spatial representation of the actual image itself, just given by the image and the blur function. Now, if you are willing to tolerate more complexity, you could do a simple thing like I did, where you make a few of these diffraction limited spots and scan them. And then you just get essentially an integral between these spots projecting onto the signal. You can do many, many other things, and many other people have. So there's many, many other variants, each with their pros and cons. So probably a lot of you know about Naji and this volumetric imaging of the brain at synaptic resolution. So she generated a Bessel beam. So these Bessel beams, instead of doing multiple spots in space, you do something that looks like a pencil or a pen of light. So this then projects all the signal along this long axis. So it integrates, instead of a discrete spots in space, over an extended slab in space. David Tank has done some work using multiple spots laterally that have tilt to scan. It's called V-twins. Kasper Podgorski, who's at Genelia, has used a technique, both the SLAM and then SLAP me and the SLAP 2 microscopies, where you do a projection of essentially multiple spots and integrate them. Ali Pasha Vaziri has used these wide temporal focus things, and he just had this preprint yesterday that he says he's imaging like 200,000 neurons at high rates of speed. So, many people are using different variants of these, but they all have the same approach, which is to say, how can I shape the point spread function and use information about my sample to design an imaging system that extracts out the features that I want? So we call these things these task-based approaches. And I think this is a really powerful thing in microscopy. I'm going to lower my shades again so I can see the screen. So look, these fundamental approaches, I think, are really have revolutionized what you can do in terms of largeness of signals and neurons collected at a certain rate of speed. And I think it's important to realize that there's many of them, because any one of them, like the one I talk about, might not be the one that you want for your experiment, right? So you should know that these things exist, but just be aware of the general style of all of these things. So I'm going to skip over a slide or two. We'll just get rid of this one and just say that using things like anatomy as priors, also something that Cinda, that you had mentioned, talked about, or brought up in your question, can really, really help our ability to take images. So this is a paper published by one of Andrea's former colleagues, Johannes Friedrich, with me and Liam Padinsky and some others. And we basically just analyzed images and said, like, images, they have a lot of redundancy in both time and space, especially if you're doing calcium imaging. So what you see, I think, is this a movie? It's not a movie. It should be a movie. Anyway, well, what we did here is, this is a really messy, ugly picture, but we have data. And what you see are these kind of CNMS extracted sources. That's the raw data on the top. And if you do CNMS, you can get the noise transient. So if you look from the image at the top, the top left to the middle top, you can see it got rid of some like little speckles in the background. That's a denoising. And if you look at the residual image, you just see basically noise. You don't see any signals that look like neurons, right? So there's I think the neurons like right there, and like a neuron there, the denoise, and you see in the residual, there's nothing there. So what happens if you actually took a lower resolution image of this? You'd see something that looks like the image at the bottom. Instead of seeing noise and instead of the neuron, you can see this neuron overlaps its boundaries. It has a lot of pixels because it's low res, so it no longer fits to the spatial boundaries. But, oh, there's my movie. It's actually working now. Great. But what we showed in this paper was, if we had a high resolution movie to begin with, so if we took like a normal slow movie, we could then go back and take a lower resolution movie just with a few pixels, basically many, many less pixels. In that case, it's 256 times less pixels. We could generate a movie that we could see signals, and using the prior of the high resolution anatomy, we could essentially localize those signals very well to the true anatomy. So this was another method where we used a multi-phase imaging approach, where we said, well, if we have an idea of what the anatomy is, we don't have to bother spending a lot of time generating those details, and we can just care about what we want, which are these fast functional signals. And then we can just use localization to do a best fit to apply it to the region that we care about. So all of these tricks lead to increases in imaging speed and reduction in processing time. So lower pixels you have, the more prior information you have, the better all of your methods are going to be. And again, these have been used by lots of other people, very similar strategies with very similar algorithms to do these kinds of all of these kinds of methods. So this is, I think, in the last 5 years, a new style of imaging that's become very powerful for doing functional recordings. And just some examples. There's a bazillion of them. One that I use more than others of all these methods, I use kind of some of my own, but I use Nagi's Bessel beam technique. And for this technique, again, for sparsely labeled things, if you look at this movie in the lower panel, what you see is a Z-stack. This is a Gaussian Z-stack of a normal signal, and that's a maximum intensity projection. And then what you see is, this is just the single beam, actually Bessel imaging. So now you're going to see a functional activity recording of this neuron, right? These dendritic processes are spread out all through Z, but there's only one label. So if I try to image it with a Gaussian beam, I'd only see part of it. If I care about dendritic computation and I have sparse labeling, Then what I want is a really long sampler that slices through the volume and lets me see the whole dendrite. So how many people have used one of these tricks in their normal imaging or thought about them, like Bessel beam imaging or anything like that? Any of you guys used it? Anyone care about dendrites? Or axons? You know, axons are sparse. There's not so many of them in a small thing, right? Techniques, I think, can be really a good way to go if you have something that's inherently a sparse signal and you want to see a lot of it. Making these kind of pencil beams lets you interrogate their large volume very, very well. So we use actually these Bessel imaging techniques quite a bit, and we find them pretty effective for that kind of thing. And again, using an anatomical prior, that can help supercharge the signal extraction. So I think these things are really, really nice ways to do imaging if you have a problem that's well matched for it. And, you know, apparently I was not paying attention with my animations, but that's essentially what it does. It scans over everything. So you can do a lot of stuff using these different projections. And you can think of these in the last, these are all papers really in the last year that have really enabled large scale functional recordings over many more neurons than people previously thought was possible. So I would say, go as far as to say these methods have kind of spurred a revolution in functional imaging, at least in mice, where we can be pretty invasive. So this preprint from Ali Pasha that just came out, I think they claim they imaged about 1 million neurons at a few hertz. So in cortex, so it's quite impressive, you know, in these tools. So just a few years ago when the Brain Initiative started, people were saying, if only you could record like 10,000 neurons. So it's really, you know, we've reached a point where in a few years we're at least theoretically, you know, a couple orders of magnitude higher than the goals at the beginning of the Brain Initiative. So I think a lot of these methods have been really been quite impressive. But one thing I want to remind people of, because I think it's important when we talk about imaging technology, is why do these methods work fundamentally? Like I started thinking about and doing these experiments like 10 years ago. The paper wasn't published till, you know, let's say 4 years ago, and I wasn't the only one thinking about it. But what's really changed in imaging that's allowed these techniques to be really good? There's two things.
Is one the indicators that we have now that allow us to spend less time in each region to get a good signal?
Yeah, that's exactly right. And what's the other thing?
Faster recording systems for data acquisition?
You know, I mean, they've gotten cheaper, but they haven't actually ended up getting much faster. I would say it's work like Andrea's work, which is trying to come up with algorithms to do efficient extraction of the signals once you get them. So I have this quote from a famous person back in 2016 that does, you know, one prerequisite for data analysis in the context of multiplane imaging is that there be sufficiently sparse activity in low background. These conditions are met by genetically encoded calcium indicators, such as a GCAM6 family. And then with conventional dyes, the background is always bright. When we saw that GCAM6 started out dark and went bright, we realized this would give us the effective sparsity we could exploit. So almost all of these methods are really enabled by the improvements in the sensors. And that's a big thing to note, that you know, the sensors go a long way in what you can do. And you know, we sometimes ignore it when we build our imaging systems. We talk about it as if we're the impressive ones, but really making the good sensors is what really helps these things work. And you know, to go back to this counting noise and how what kind of having good sensors gets you, right? When I first started out doing calcium imaging, you had like Oregon green, OGB. You loaded cells, and that maybe had a 10% change with action potential. The signal was bright, and then it got changed color or changed intensity a little bit. So, you know, if you have something that would have a fractional modulation at 10% activity, you'd need 1,600 photons collected to detect it with a signal to noise of 4 over something. So that would be like, oh, if you have 16, you have 16, the signal is collected as n, the standard deviation of that is the square root of n. So to get that number, if you have a 10% change, to have that 10% change be much bigger than the square root of n, I needed 1,600 photons. Doesn't sound like a big number, right? I mean, but if I have to get that for every cell, it could be. Now, if I go to GCamp, which has like 100% change in signal intensity, I could, in theory, do that with 16 photons. So if you imagine what my imaging requirements are, if this was in photon dwell time, I just changed my dwell time by a factor of 100 in order to get that signal and be able to record an action potential. So almost all of these things rely on good sensors. I want people to remember that, right? Good sensors are the key. I should say, fancy tech can't turn garbage to gold, right? If I had crappy sensors, no amount of these imaging methods and algorithms would be able to turn it into something that's great, right? So, it's important to keep track of these sensor developments. And there's some imaging techniques that may not work for you now, but with just a small tweak of the sensor, it will become very, very powerful. So just pay attention to what's going on in the sensor space. It's really important. So, with that, I'm kind of done my imaging summary. And the key take-home messages I want to say are, right, one size doesn't fit all. So we have a variety of strategies from kind of vanilla two-photon imaging to these approaches which use multiple beamlets and complex projections of pixels to extract out the things. The right one for you is the one that matches the questions that you're asking on your sample. If your sample is very dense and complex, you might not be able to use a crazy imaging method that relies on projecting many pixels into one space. The other thing is we can gain by shaping the things we can control. So by changing the point spread function, we can generate all of these imaging methods. They're essentially a normal two-photon microscope where we just change this point spread function, which lets us know how we interrogate the sample. If we know all these things, we should realize that all imaging involves some kind of estimation, right? We're recording signals from a blurred region in space and time. So we should take advantage of these statistics when we develop algorithms like the spatial unmixing method that Andrea talked about. And we should leverage prior knowledge of the sample, like the anatomy, as much as we can to generate these signals. So together, using all these kind of design criteria now we can build systems where the hardware and software is really built together to create imaging systems that I think are much more powerful than imaging systems of a few years ago. And I think it's a pretty cool thing to see all these changes that have happened in the field. Before I move on to modulation, does anyone have any imaging questions for me or anything that I went over a lot, so.
Yeah, hey, Darcy.
Sorry, later. I'll go on.
Yeah, I have a quick one. So the some of the imaging techniques you showed, they are not super trivial techniques, and there aren't that many labs that have the capability of doing those techniques. So when do you think the gap between everyday neuroscientists who performing experiments but do not have like a deep understanding of how microscopes works can use some of these techniques, you think?
So I think it varies very much with the technique, with a specific technique. Some of these techniques are being or have been commercialized, or there's small niche companies that will make like a module to go onto your microscope. And there's even some companies, there's a guy named Bruno Pichlar who has a small company. I always forget the name of it. Is it INS Spencer or something like that?
INSS.
INS.
Independent Neuroscience Services.
Yeah, that could help people implement these things. Now, my feeling is, so I teach at a variety of places like Cold Spring Harbor and Woods Hole and other places. I think the best way to learn about these systems is to visit someone's lab who's doing it and learn from someone who's actively building a system, and ideally do an informal collaboration with them first and see if the system works well for your prep, right? Before you want to invest a few $100,000 in fancy lasers to do technique, whatever it is, it's nice to know if it would work in your prep. That being said, there are some other methods like the Bessel beam imaging that, you know, you can kind of implement them for relatively low cost as long as you have access to your microscope. And I think the decision should be just based on, one is a critical read of the method and comparing that to your experiment and say, is there something that if it worked would really even help me answer my question in a meaningful way? Or would it just kind of look nice on my view graphs, right? And that's a legitimate question, I think, to ask a lot of times. And then the second is then try and find a local friend or a collaborator who wants to disseminate it. And I'm going to put I'm going to throw shade at lots of developers or not, and just say, if a developer has a new method and they want to see people implement it, I think there's a responsibility on that developer to either try and provide training or materials or kind of workshops to help get it out there. And some are very good at that and some aren't. So I would say, you know, us as a field, we have to hold methods developers in a sense to task in our dissemination efforts to try and get that to be spread. And I think, you know, Spencer is, I think, actually a good example of someone who shares broadly through both these workshops, preprints, and like lab rigor and other type of things. So just, you know, put a fire under our feet. That's what I'd say. Come and ask us questions, and we'll try and help you to evaluate that if you have trouble. You know.
Thank you. Yeah, it's one of my favorite things to do. If people send me questions, I try to get back to them as soon as I can. Darcy already mentioned Bruno's company. There's another company that's similar. Dale used to work with Bruno. Dale now has his own company, COSYS, C-O-S-Y-S. I'll put them in the Slack channel if anybody wants to reach out to them. But it's a great. Basically, you're hiring like an Uber postdoc for, you know, getting something running in the lab.
Yeah, they're really awesome. I can vouch for them.
I have a quick question, Darcy, a comment for everyone, actually. So, sorry, I go back to this point of real-time system, but I think big opportunities might open from the fact that maybe now in real time you can know what are the neurons, right, that are active in your field of view, how much is their signal-to-noise ratio. Are you aware of any work that might exploit this? So, I mean, now your optical system can actually adapt in real time to what you're reading.
Yeah, so we do some of that. So I have a small project that, I mean, you know, started a long time ago when I first joined Rafa's lab, which was to try and do voltage imaging with spatial light modulators in 3D. So in that case, we do a scan, we then generate beamlets where we target individual somata, and then we extract out signals. And we change the intensity and the targeting based on what we see in real time. And we have things that are kind of, we call it like attentional imaging, where if we see a change in signal in one area, we move to another or we interrogate it more. So we do some things like that. On the more, let's say, technical front, I mean, Casper's latest technique, and I mean, and he's not the first to do it, but in this implementation, right, he uses this SLAM2, where they use a DMD. It's another form of a spatial light modulator, you know, using mirrors that can tilt on and off to generate spatial masks of the regions of interest you want to illuminate. And then you use a really powerful laser to essentially do kind of like almost like wide field illumination that then is a mask to only target your regions of interest. So that's an experiment, again, that's using selective illumination to lower the photon load, the heat load on your sample while targeting in real time the things that you care about. The further extreme for that is Chris Hsu is developing, so I'm not going to talk much about 3-photon lasers, but you know, 3-photon is a technique that you can see a little bit deeper, you have enhanced contrast, but at an expense of a lot of things like laser power, all kinds of variation. So Chris has made an adaptive laser source that you take the first image of the conventional, so to speak, image, and then he has a laser that he can turn on and off between the neurons. So he only illuminates the neurons of interest. Now under 2-photon microscopy, this isn't bad. You can use a Pockle cell to modulate it on and off. But for 3-photon imaging where you need super powerful lasers, it's not as efficient. So he's called this like a pulse on demand system to do that. And I think all of these could use good integration into real-time controls and extraction of your signals. The ideal case, what I would like to do, and probably other people too, is I kind of want to control the illumination based on the SNR of what I sell. If I have a cell that has really huge SNR, I'd like to actually like lower the intensity on it to just give me sufficient SNR so I don't bleach it or heat it up. So I think there's lots of opportunities in that space. And I think Bruce might talk about some of those because I know, Vidrio, I used to talk to them a little more than they have since COVID, but I know, well, from my discussions with them, and I know they're talking with lots of others about ways to implement these things very well. Maybe he'll talk about that tomorrow. So anyway, if that's good, I'm happy to answer questions in the chat and Slack too, but I'm going to move on to the next part of my talk, which is on kind of modulation steps, right? I've talked about this imaging and now modulation. And, this is kind of the dream, right? We'd like to be able to perturb neurons and get the mouse to change its behavior. And we'd like to change its behavior in a deterministic way. So, my favorite plot of this, I made it from, a normal kind of bifurcation diagram from, one of these chaotic systems, which is, the mouse starts out in one state, and then at a bunch of different times, there's a series of choices it can make. It can make a bunch of computations, behaviors, whatever, and do something. Well, we'd like to be able to control the trajectories of these neural dynamics that we can essentially drive it along particular states. And that's kind of our dream. I don't think we're ever going to be able to control a gazillion neurons at once, but we can already do quite a few at once. And, I think this is really powerful. So I'm going to talk about some of these methods. Give me a show of hands or some shout outs, some yays or nays. How many people here do two-photon optogenetics in their home lab? Or want to?
Yeah.
I think a few people want to. I mean, presumably, if you don't want to, just bear with me, but, generally, right, we'd like to be able to control neural activity in some kind of meaningful way, right? And, for me, this was kind of like the first time I imaged a mouse brain and saw the neurons fire is, right, one of the things that optogenetics gives us, and specifically two-photon optogenetics, is we don't want to just define cells based on their genetic identity. We know these cells participate in different computations in different ways, right? So when I look at some, let's say, interneurons or pyramids, that's not enough to know, like, okay, I want to just excite all pyramids at once. I want to be able to basically just choose any cell I want based on what it does at a particular function on anything and use it to shoot. So there's an experiment where we were first trying out where what you see in the circle is essentially, this is like a bullseye. This is where my two-photon photoactivation laser is firing. And what you see is in the red, it's basically, it's just some red indicator that's, really poorly expressed. And then in green, you'll see basically GCamp when it lights up. So if I look at this movie now, I moved the target off of the cells, and you'll see those lines in the background. I'm photostimulating something that doesn't have a cell with opsin. And now I stimulate that cell, and you can see they have the big calcium transients. So, with these kind of techniques, you can literally just go and draw circles on neurons at your leisure and get them to fire. And, the first time you do this, it's cool. It's like you can control something. It's pretty nice. Now, just controlling one or two neurons is not going to get you really big, meaningful results now. So what we want to do is we want to control lots and lots of neurons. And, Nico talked a lot about that previously. So, this is work, again, out of my lab. There's many other people who have done similar work, where we combine optogenetics with imaging so we can simultaneously image. This is Wei Zhan, again, back when he was a postdoc, now again, a professor at UC Davis, leading his own lab. And what we have here is we have a conventional normal microscope, and we basically add another arm that has a spatial light modulator on it and a fancy laser. And we can now essentially control both imaging and photostimulation separately. And with that, then what we can do is we can do the same tricks that kind of Nico was showing. This is a slightly different method where we generate these beamlets, very similar to what we do in multiplane imaging as well. And we can simultaneously target multiple cells in 3D and get them to fire. So in the field of photostimulation, there's a lot of discussion by aficionados on how do you stimulate, whether you stimulate by shooting a cell with a pancake or a shape, or whether you shoot them with a point that you kind of paint over the cell. Either one works, I should say. In these experiments, we're doing this painting method where we have a single point, or we have a single point going to each cell, and simultaneously we paint over each cell. So you can do this and you can generate patterns in 3D. This is patterns in the XZ plane. You can see we have spots that are elongated. We can simultaneously target, 100 cells. This is now in targeting it in a fixed fluorescent sample. And what you see here as we're scanning, we burned all these spots simultaneously in 3D. So we just did it as essentially a single shot, and now I'm doing a Z volume through it. And this is similar to what Nico was showing yesterday. We can generate these disks everywhere in space. And you can see at different layers, we can generate different patterns. They actually disappear, they're confined. And because everybody at schools like it when you put school logos in, we have like SLM 3D CU for Columbia and then a smiley face, right? I mean, but you can imagine these patches are about the size of cells. And this lets us, do two things, check our targeting, whatever, but it just demonstrates that we can target multiple sites now in 3D and use it to drive neural activation. And again, I drew my little kind of funky looking spiral at the bottom because that's essentially what we're doing. We're generating essentially diffraction limited spots, and then we spin them and paint them over the cells to get them to fire. And in this paper, we showed we could stimulate lots and lots of cells. To do this, multiple targets, and it was really, really easy. And you can stimulate both loads of cells. So, I mean, this is an experiment where we're driving massive two-photon activation, right? These neurons are firing like this because I'm doing two-photon activation. It's not because it's having a seizure. So, right, we can now drive on the order of hundreds of cells when we want simultaneously with imaging. And this is kind of a cool thing. The key about these things are, though, what people were asking yesterday is how many cells can you actually do, right? I mean, I think that was a question that went to Nico, and it goes for imaging and so on. What are the limits of what we can do? And, we had some hints on this for imaging from a paper that came out of Janelli. Again, Casper, when he was developing some of his methods, it was really, he really needed to show that the amount of light he was putting on the brain weren't frying the brain. So he did a series of extensive tests where he looked essentially at damage due to imaging in the brain from just giving illumination. And under this, looking at a lot of parameters like heat shock protein, GFAP activation, all these things, he really showed, I think convincingly, at least in this prep, that you could put a few hundred milliwatts on the brain for a pretty long period of time without causing much damage. But clearly somewhere between 250 milliwatts for 20 minutes and 420 milliwatts for 20 minutes, something bad happens, right? You can see that. So these kind of set the limits are at least gave some defined criteria for how much can you get away with. And, if you're imaging and I said, I want to make beamlets where I image hundreds and hundreds of cells at once, you'd probably be thinking that you're putting way too much power in because, you need tens of milliwatts per site to do imaging. Give me a number for people who do GCamp imaging, how much power do you normally use?
I recently measured like 30 watts coming out of our scope for GCamp imaging.
Milliwatts, maybe?
Try to keep it under 100.
Sorry, 30 milliwatts.
Yeah. So that's one spot, right? So if I had 100 spots at 30 milliwatts, I'm at a few watts, right? So this makes it really hard. So what's a trick? What do we all do to be able to image like this? Well, we use these really low rep rate lasers. And if you imagine, two-photon efficiency goes as the peak intensity. Now, there's a difference between fluorophores and opsons. If I want one photon to come out of a fluorophore, I have to put one, or in this case, two photons in, right? Every time I put in two photons in a fluorophore, it gives me back one photon of fluorescence. But that's not the case when I'm opening an ion channel. I open an ion channel, and for every set of photons I put in, the ion channel just conducts however much at once, right? whatever its native conductance is. I don't have to keep firing it again and again and again. I can turn it on and it'll just keep on giving me the signals that I care about as long as its time is open for the lifetime of the channel opening time. So what this means is I don't have to rely on imaging where I have to kind of keep pounding the fluorophore, the cell, or the opsin to get them to open. I can essentially just turn them on just once in a while and they stay open for a long period of time. So to do this, if I change the rep rate of my laser from 100 megahertz to 1 megahertz, but I keep the average power the same, that pulse at 1 megahertz has 100 times the intensity, right? Is the average power the same. Now I do 2-photon of that, it has 10 to the 4, it has 10,000 times more 2-photon peak intensity. So if I move to low rep rate lasers, I can actually still have very, very high powers that do very, very efficient driving of 2-photon excitation with having the average power being really, really low. And if I had a normal fluorophore, this wouldn't help me because pretty soon, if I only shot it once every let's say millisecond, I'd only get 1 photon. But if I have an opsin that the channel stays open for a millisecond, it basically is open all the time, even if I only shoot it rarely. So this was a really big thing where we could show that we could essentially fire cells with kind of less than a few milliwatts per cell. And under these kind of conditions, then being able to stimulate hundreds of cells becomes kind of possible. Because I'm taking advantage of the characteristics of the light and the biophysics of the opsin, to say I just need to turn it on and I don't have to hit it again for a long time. So I can now switch my whole imaging strategy or laser strategy from one where I would need to give it pulses each time I get a photon out to one that I just basically ring it once in a while and I wait for it to become quiet again, then I ring it again. I don't have to keep tapping it. So this was a really, really important thing. And again, a good thing for doing imaging. So when you see a lot of these methods, you'll see the people shifted to these low rep rate, high peak power lasers for doing imaging because of these advantages. And one thing I should note about all these things when we do opsins is most of these opsins also absorb at the colors where you're imaging. There's crosstalk between the opsin and indicator, especially if you're using GCamp, which is kind of the best indicator for calcium anyway, and the red opsins. The red opsins all absorb a little bit. So if you are doing these experiments, I should say, hey, this last panel here, people should be watchful that when you do imaging, if you increase your imaging power too much and you have an opsin, you can see the firing rates increase with imaging power. So for all of these kind of opsin experiments, you have to watch out and use those controls. You could do a different strategy, one that Tommaso Fellini at the IIT in Italy has used, which is you just go to the blue opsins, like the normal channel red opsin 2 or those ones, and then you image with red indicators. But while the red indicators are getting better, for us, usually the value is still using GCamp or the like and not the red indicators. But you can use that strategy to avoid that kind of crosstalk. Okay. So now that I hope I convinced you that at least we can stimulate cells reasonably well. One of the tricks we can do it on power. So, what do we like to try and do? And this goes to Andrea's experiments. We really want to do closed loop ensemble imaging. So what we want to do is we want to actually fire cells as a function of their neural activity, or of those cells or other cells. So we have all kinds of strategies to do it. This just isn't meant to be anything big, but it just is representing kind of a paradigm that we do is we essentially do a baseline calibration. So we watch neural activity to figure out what cells are doing what. This is kind of the measurement step of the closed loop. And then we do some analysis to do some mapping to figure out which cells do we think are doing something interesting. And then once we have that, we enter it into a closed loop paradigm where we say, okay, in this case, we pick two ensembles, E1 and E2, their names are arbitrary, and we say, we want to activate one ensemble, but only when the other one is suppressed. So in other words, we want to set up the system where we can kind of wait around, and only when certain neurons are silent do we activate other neurons. And, we basically do these kind of things. In this case, it's for a brain-machine interface experiment. But now we can get these things to work and update on the order of, let's say, 60 hertz. So it's not as fast as what Andrea was talking about, but it's generally decent enough for some of our calcium imaging right now. So we do these kind of things. So what's something that you think maybe I would care about when I'm doing stimulation? I just told you I did single cell stimulation, but I don't think I showed you any data that really demonstrated it. Did I? Do you believe I can stimulate a single cell? And only that cell? And no other cells? Does it fire every time I shoot it? What do you think?
Can you kill all the other cells first?
Yeah. Yeah. Do you think I need to do controls?
Well, now you have calcium imaging, but it's difficult, I think, for you to, since you don't know that, one pulse will activate one neuron, right? There will be an integration time. So it's difficult to know, to calibrate precisely the spike, right?
Yeah, it's super hard. So, like when you read these papers, everyone claims, and, like even people like me, you're like, oh yeah, we stimulate these things all the time. Be skeptics, man, be skeptics. That's what I want to say. So, we're skeptics ourselves. So I force all the people I work with to do a bazillion controls. So this is work done in collaboration with Rui's lab, Rui Costa, and then a little bit from Jose Carmina's lab at UC Berkeley, where we're using a new opsin, or a relatively new opsin from Carl Deiseroth called Carmine. And, I basically like, I should say, I'm going to use the words I force them, but I don't force them. They do these on their own things, on their own accord. But it's more fun to pretend that I have control over what they do when the reality is not true. But, so I forced them to do these experiments. So we did a few hundred sessions over multiple mice, hundreds of stims per cell, looking at longitudinal stability, all of these things, whether it was clean control. And when we do these, when we pick these cells, we can show that on like every single instance, when we do two cells right next to each other, right, we stimulate the red cell. That's the cell we want to stimulate that we're targeting. The cell next to it, basically the green cell, that's just an adjacent cell that we don't want to target. Every time we fire and try and stimulate the red cell, you see we get a transient, and we never get a transient on the neighboring cell. And it's not that that cell is not active. If you look at that kind of raster plot above it, you see it as spontaneous activity. And when we do choose to fire it, we can get it to fire. So we sit around and do tons of experiments like this. We pick some other cells that are close by and we try and do that. So we spend a lot of time trying to figure out these maps, to figure out what in fact is the experimental resolution in the prep of interest. And this depends on the type of cell type that you're doing, the region of the brain. And that's because the opsin expression is a little bit different, and the just intrinsic excitability of these cells is different at different times. Hippocampal neurons have different excitability than the pyramids in the cortex, and layer 2-3 is different than layer 5. So we actually spend a lot of time trying to figure these things out. The same thing goes for trying to generate.
How do you know, Darcy, that these things aren't related to just the neurons being somehow connected slightly or something? I mean, maybe there's, some fire, stimulating one cell might cause some effect as opposed to the background from, laser power exciting the neighboring neuron. How do you how do you control for that?
Yeah, so that's a great question. Usually, I mean, there's a few ways we do it, all of them somewhat indirect right now. So one is we can look at the timing, the latency of the activation. Now, with calcium, this becomes hard, right? Because the calcium rise times are relatively slow. So to see a monosynaptic connection, let's say, would be very, very difficult. But from what we generally know about synapses, at least in cortex, the inputs from one neuron are insufficient to drive another cell to fire. So in cortex, what we can do is we can basically just look at the reliability of activation and come up with kind of histograms that let us have some idea of what the conditions are that cell may or may not be activated. In other cases where we see a delayed response, and we certainly do in some neurons, we see a latency before the firing. So, instead of basically seeing the rise happen within a few milliseconds of our activation, it might be 30 or 40 milliseconds after. And we do see such Fowler neurons. Ideally, one would have voltage imaging, and then you would just directly map out those processes and see what those things are there. That's not yet, it's not yet there. The other thing that we do is in most of our experiments, we're not just stimulating one neuron, we're stimulating multiple ones. And what we do is if we see ones that are connected or seemingly connected, we do a lot of controls where we keep the number of neurons stimulated preserved, but we might do dropouts of any cell. So it's kind of, you know, we have a pattern and we just shift 1 or 2 cells in that pattern and we see how it affects the activity. But in generally, if the activity is driven strongly and it's very fast, it's very hard to disambiguate those two things. So here's just a map of our general spatial resolution in these things, right? You can see the hotspot. This is looking over a cell. These little spots are 5 microns apart. And you can see the calcium transients on the PSTHs on the plot, and then the effective target response versus the distance to the centroid of that cell. So in these cases, I think, you know, this is reasonably convincing, right? That when we target a single cell, that cell is really firing with pretty high specificity. Now, this is a different statement than saying no other cell is firing. So I want people to realize that, right? The statement I made is I can reliably fire the cell that I'm targeting with high spatiotemporal precision. That is not saying that a nearby cell may not be activated if it has some processes nearby to do it. So these are distinct claims when people are making photostimulation experiments. So, you know, I think people should really think about that, because a lot of times people will show a plot like this and say, use this, and then say, and therefore I'm only stimulating my cells that I care about. Well, this plot doesn't show me that. This plot shows me that when I target a cell I care about, I get it to fire. It doesn't necessarily show me that every cell that I don't care about is not firing. So I just want people, when they see plots like this, to think of the converse, right? This is showing one side of that specificity, not both. So anyway, when this works, right, we can use these things to drive spatiotemporal patterns. Here's just a stupid example. I just like them, right? We basically drive a sequence in these 4 cells. One's a little bit dim. It's not super exciting. But, you know, like I like showing videos of actual signals, what it really looks like. And what you'll see if you kind of follow the arrows around from this cell to this cell backs up, it makes this loop. So now we can kind of control these things. This is using a somatic restricted opsin. So the opsin's just on the neuropil. And, you know, when we actually look at what it does, is if we look at the calcium transients, we can show we can reliably get these cells to fire in the order that we want when they want. And, you know, it's not a super powerful experiment. We're only doing 4 neurons like this, mainly because it's hard to, it makes a nice looking demonstration. But we can do this for now many tens of cells to hundreds of cells, where we can deterministically get them to fire in whatever kind of spatiotemporal sequence that we want in the field of view. And now we're using these to kind of try and drive ensembles and do actually behavior experiments where we use this to modify a behavior. So this is kind of the, you know, let's say the state of where we are on these kind of experiments now. And just I threw in a slide while we were talking to show Bruce would address his question. Here's an experiment where we were actually trying to stimulate certain cells and not stimulate other cells. So we had ensembles, E1 and E2, that we were driving. This is work that was done with Vivek Athale and Nuria Lopez and Inez Vaz from Rui's lab and Jose Carmina's lab. And what you see here is, if we look at the average response relative to the time of photostimulation, so there's a top right panel, every cell that we wanted to stimulate has these big rises consistently. But if you look the second blue trace from the bottom, you see a time-lagged response. It comes every time we stimulate these 4 cells, right? So here's a question that we can see an activity, but the latency clearly disambiguates this from direct targeting. There's other cases where that's not the case, or in some more direct follower. So I just threw this in to show one example of a cell that we're perturbing the network in a way that it drives cells that are not targeted, that generates activity, but in a way that we can disambiguate with timing. You're not always so lucky, I should say. Now, when I talked about that non-specific activation, I just want to say that as you increase the number of cells you stimulate, you get increased non-specific activation. There's no way around it. I don't want to spend a lot of time on this, but just say if someone says they stimulated 1,000 cells at once, and it may be true that they hit those 1,000 cells, but they also probably hit 1,000 others. So Oh, because the more complex your light goes, the more chance that like little fragments of light or little axons or neuropil of passage will also be stimulated. So just be mindful that when we're doing these experiments, the more things that we stimulate at once, the more complex the analysis is. So that's, I think, just a key point for these, because now people are starting to do more and more experiments where they're stimulating more and more cells and making, I think, broad claims about how accuracy of targeting. So it's just something, I think, something to think about.
Hey, Darcy, I have a quick question about that.
Yes.
So Carmine is supposed to be somewhat restricted. I'm wondering, related to like what you've been talking about, have you noticed specific cases where you've like noticed Carmine going into the dendrites and you're stimulating and it seems to cross over those dendrites?
I would say if using the somatic restricted Carmine, I know we see it's reasonably tightly restricted to the soma and some immediately adjacent perisomatic region. So it does creep up sometimes on like the trunk of the apical and the obliques, depending exactly on the somatic restriction sequence used. But on the ones we use, it looks like it's mainly in the soma with some little puffy cloud, some little leakage just in the perisomatic region. Now, what happens though is if you imagine, because I don't know what the Rio base of any one cell is, it could be the case that if I'm stimulating an adjacent cell, or I could still get the cell to fire by just stimulating, by just having a little bit of light on those perisomatic regions. And that can especially be true in vivo when you might have, during a behavior, when you might have other inputs driving the cell near threshold, and then the opsin is just giving essentially another few effective inputs and helping push it over. So those are the cases that it's really kind of hard to figure out what's going on when it's kind of the opsin alone is not enough to drive the activity. So if you directly targeted that under quiescent conditions, nothing happens. But under your experiment, when they're doing the behavior, the cell is biased or depolarized in a way that it can drive excitation. So those are things that we try and worry about and think about when we're doing our own experiments. I don't know if that answers your question.
Yeah, I guess over time it might creep into the processes as well. I mean, we're using Carmine too, and I think over time we do see some of it in the processes.
Yeah, I mean, I'd say we've looked at some mice maybe after 4 or 5 months of expression, and some of them were good and some of them were like crufty. That's only way to say it. So I think, yeah, it depends a lot on the mouse. And, we've been playing around with different, either the bicistronic vector with GChem, so it's dual virus, or even we see differences in that one versus just a packaging that just has the opsin into a transgenic line. So we see some differences even in exactly the nature of the cassette for the expression.
Cool, thanks.
Yeah, and I would say one thing that let's see, I'm going to skip over the next slide just because I can just show this one, which is I think good to note that one reason why resolution isn't always as good as we claim it is with the opsin is kind of related to the thing that I talked about earlier, which is we use these high peak power lasers. And these high peak power lasers can easily generate enough intensity that they do what's called a saturate to two-photon transition. And that means like the peak power is already high enough that everything that could absorb does absorb, right? So if you imagine if that's the case, let's say I need a intensity of 10 to get an opsin to absorb. If I have a Gaussian and the peak of the Gaussian is 10, only the tip of that will absorb and I'll have an effective resolution that looks good. Now imagine if I just jack up this whole Gaussian, make it bigger and bigger. The shape of this Gaussian hasn't changed, but it's so much wider where it's over that arbitrary number 10, right? It becomes much, much bigger. So by using these high peak power lasers in the process of this photoabsorption, I can get these saturation modified point spread functions, which then means even though my point spread function, I think it looks good on paper, in practice, it grows laterally and axially. So my targeting changes depending on how much power I put in my sample. So in other words, that resolution plot that I showed you is a resolution at just one particular stimulation condition. It may be different and worse as I increase the power. So that can sometimes happen and change your experiment. So same thing, if you want to start doing these experiments, make sure you do your calibrations at the powers that you actually plan on doing it, because I could game the system very easily by turning down my power and titrating it so everything looks great. But then I do my experiment and I turn that volume knob to 11 so I get signals, and all of a sudden I'm in a regime where my effective resolution is much, much worse. So these are just things from the practical basis. I think, whether it's imaging or photostimulation, people should realize when you do your checks and stuff, the actual conditions you're doing the experiment, if they're different from how you did the controls, the results aren't necessarily valid. So, this is an important point, especially in photostimulation. So I'm going to skip a slide and just say that, these algorithms that Andrea and others are developing, I think, are critical to do these real experiments, because, in our experiments, getting the data is essentially the easy part, right? It's figuring out which neurons are active, do the extraction of activity, circuit inference, make predictions, and then driving those things on photostimulation. That's actually really, really hard. So in some sense, the systems that I work on are just the front end of these systems, and really relies on good collaborations with both groups like Andrea's or others, and people like Bruce, who are designing microscopes and microscopes control software to do this really, really well. So if you want to undertake some of these experiments, then I recommend don't do it alone. Reach out to your friends and neighbors and try and build teams to try and address these really big problems, because it's really hard to do well. I'm almost done. I have some more stuff, but this is the conclusion for the controls, and I wanted to say to everybody, it's clear that we can do stimulation very, very well with algorithms that do holography, like the ones that Nico showed, the ones that I showed. We can target huge volumes of huge numbers of cells in 3D with high spatiotemporal precision. We can do this in combination with imaging now, which I think is really, really powerful. And we can identify the cells by genetics or function. So to me, that's probably the most powerful thing about these approaches is I don't have to rely on intersectional strategies to label them. I could just observe a bunch of neurons, find the ones that I think are interesting, and then perturb their activity. And I think that's really necessary for doing mechanistic controls, so we can actually try and sort out causation rather than just correlation in neuroscience. With that, I think, I should say, great power comes great responsibility. You really need to have good experimental design. Otherwise, you do a bunch of joke experiments. Now, I'm going to show a thank you slide, but then I'm gonna thank everybody, but then I'm gonna go on to something else, because people were wondering about grid imaging, and I made some grid imaging slides for this. And Spencer, I have some time still, right? A little bit? Spencer's probably out, but as long as you guys don't say no.
You can take a little bit longer, Darcy, it's fine.
Yeah, okay. So I just want to thank everybody and thank my collaborators and stuff, because most of this work is, not done by me anymore. I try and help give some guidance, but it's really the work of a few people that I kind of share and co-mentor with Rui, Inej Vaz, Vivek Athali. I do a lot of work with them. They're working on almost all the grid lens imaging and photostimulation stuff that I've been showing. Great collaborator with Liam Paninsky and Rui for a lot of stuff in the lab. And then there's a lot of other people who I showed some of their data, old work when I was with Rafa and Weijian, Estikios, the partner in crime of Andrea, and Money from Brain Initiative, whatever. And then here's our fancy building in New York. I want to make sure to thank them, because they're the ones actually doing the hard work, not me. I just get to talk about it. So with that, I'm going to move on to some GRIN lens imaging, because I think GRIN is kind of a important thing, especially for a lot of people's